So today we're going to go a little bit further on that trajectory where we will consider covariates, both genetic and non-genetic covariates, and how we can use them in QTL studies. So the basic overview is something like this. I will give an overview of how we can think of covariates and so-called mediators in QTL studies and how we might adjust for them. Then we will discuss genetic relatedness in segregating populations and how we can adjust for genetic relatedness in genetic studies using linear mixed models. So what is tying genetic relatedness is that you can think of to covariates is that genetic relatedness is like it's a form of genetic covariation. So in that is how covariates and genetically related populations are tied together. As I think most of you know, the main idea about genetic or experimental crosses is that if you have two strains with showing a consistent difference in a phenotype in the same environment, and the genetic difference must be genetic, and by crossing, you can create a population of approximately randomly shuffled genomes where genetic variation is segregating, and association between phenotype and genotype may be interpreted as being in causal. There are many different kinds of genetic crosses one can make. For example, in rats, you have F2 crosses, which may be where you cross a strain A with a strain B, get an F1, cross the F1s with each other, and you get a mosaic of the parental genomes. This is an example of a two parental cross. You can have more complex crosses where you have multi-parental, multi-generational crosses like this heterogeneous stock, which is also a mosaic of the sort of 8 parental genomes. So the essential idea in QTLs is that we have genetic markers that we can observe, and there is a unobserved QTL that we want to detect that is causing a phenotype. The meiosis is randomizing the state of the QTL as well as a marker on the same that is located near that QTL. The QTL per se is not observed, but or neither is the kind of the meiotic event. But because the QTL and the marker share the same meiotic event, this causal connection between the phenotype and genotype is observed as a correlation between a marker and the phenotype. In subsequent diagrams, I'm going to just assume that we have a marker that is close enough to the QTL that I don't need to have this extra arrow. And I think this is a theme that will be developed further by others later in this sequence of seminars. In the previous lecture, we discussed how we can have sex as a covariate. So if you draw out the causal connections or the causal relationships and the role of sex in a phenotype, if you have a QTL on an autosome, then you have separate recombination events that are that will be causing the sex, that will be an event on the X chromosome, and there will be different recombination event that will be causing or approximately randomizing the QTL, which is causing the phenotype. In this type of situation, you don't have a direct connection between the sex and QTL. So it is, unless you're looking at a marker on the sex chromosome, if you don't adjust for sex, it is okay. It is not fully efficient, but it might be okay. So, but if I simplify the picture before, it will look something like this. So in this situation, unless the QTL is on the sex chromosome, for which we have to approach it differently, also discussed in the previous lecture, it is okay to just look at, search for QTL by just looking for associations between markers on the autosomes and the phenotype. You get more efficiency if you adjust for sex. In the same way, if you have a QTL on a different chromosome than the one that you're looking for, it's the same type of situation where if you don't adjust for QTL on the other chromosome, you would be you will get an approximately unbiased estimate of your QTL effect. However, you will lose efficiency if you are not adjusting for QTL on these other chromosomes. So in general, the idea for QTL analysis is that it is, you would search, at first when you have no information, for QTL on all the chromosomes. And as you find more of them, as you find some of them, you will want to adjust for those you find on other chromosomes in order to get a more efficient and less biased, if they're on the same chromosome, estimate of the QTL effect on the phenotype. So here is the example data that we used in the last two examples.
Can I ask a question?
Yes.
Is this okay to ask questions? I'm sorry, I don't want to interrupt.
Yes.
But doesn't
sure.
Doesn't this imply or assume that QTL is not 2 is not in the causal pathway? Maybe you've covered this previously, but you wouldn't want to adjust for it if it were somehow in the causal pathway, would you?
It is not in the causal pathway, as you can see from this diagram.
Okay. I see what you're saying. All right, thank you. Sorry to interrupt.
And I mean, if these two were on the same chromosome, then it's a little bit different. So let me see if I can do that. If it was like this, then that is, they're on the same chromosome, then you will want to adjust for both of them. Does that make sense?
Yep, thank you.
Yeah, so and in fact, this is an issue with linked QTL. It's well known that if you have linked QTL, yeah, not only, you can sometimes fail to find them if you're doing a genome scan, because the other QTL may mask or certainly bias the effect of the QTL that you're looking for.
Right.
So yeah, that's so. But in this is the situation for if you have two unlinked on different chromosomes, or if for multi-parental crosses, at least in a place where they are not correlated very strongly, so that would, in a human GWAS, you may have two QTL on the same chromosome and they could still be uncorrelated. I think as your question pointed out, I think thinking about drawing out the causal diagram, I think it makes it easier to think what approach is okay and may not be okay, or what may have less or more bias. I think is helpful to think about. I mean, a lot of geneticists have intuitive idea of those things without drawing these diagrams. But if you are new to it, you might find it helpful. So the example data that we looked at earlier was the data collected by Leah Solberg and colleagues. I got it from the QTL archive, which has a list of a bunch of all the data and the phenotype definitions. In particular, I'm going to focus, just because it's easy, on two traits: glucose, that was measured after a stress test, and body weight, and it was measured at 15 weeks, and body weight that was measured a few weeks earlier in week 11. So I think the other thing I will mention is that how to do QTL analysis with covariates is going to be the same, pretty much the same approach as what you would use for analyzing sex. I think I covered this in the previous lecture. Just replace sex by your covariate, and or secondary QTL, and everything else is basically the same. But we can come back to it if there are questions or people want greater detail. So and we are going to use Carl's RQTL and RQTL2 for subsequent analysis. Okay, so I'm going to read in the data, and here are all the phenotypes, and the things that I'm most interested in right now are glucose after the stress intervention, or challenge rather, and body weight that was measured a little bit prior to it. So if our primary phenotype of interest is glucose, and we have body weight measurement, I think one might want to think about, okay, we all know that body weight is related to the metabolic syndrome, and so there is a causal connection between body weight and glucose. Now the question for QTL analysis becomes how to treat a measurement that you know may be causally connected to your trait or major phenotype of interest. In this case, I just I note that the body weight is collected before before the glucose is measured. So if there is a causal arrow, I mean, it is you could think of a latent variable that causes both of these measurements, but let's try to think of a simplified picture of how these traits might be related to each other. So one way to think about it is to think of body weight as a mediator. So this is similar to what Bob mentioned earlier. That is to say that there is a QTL that affects glucose as well as body weight. And body weight also has some causal relationship with glucose. So in this type of framework, or in this framework, body weight would be considered a mediator. Another way to think about it, or another model for it, would be that body weight is not directly connected to or caused by this QTL that is affecting glucose, but it is controlled by a separate QTL on a different chromosome. So these are two slightly different models. And of course, you can empirically find out, at least to some extent, whether it is this or that. Because if it is this, then the same QTL would be found for both traits. And if it is this, then you would have different QTLs for the two traits. But in both cases, the causal arrows that are unchanged are the arrow from weight to glucose and the arrow from QTL to glucose. So that I'm not questioning in this model. So what are the analytic considerations? Well, the first thing is whether you are considering the body weight as a mediator or an independent effect. In this case, you can find out with a genome scan for both of these traits, whether it can be considered as a mediator or an independent effect. But in general, if you ignore body weight, even if you know that body weight has an effect on glucose, then your QTL scan for glucose is a QTL scan for the total effect of the QTL on glucose. So even in this example, if you ignore weight and just search for all associations between possible QTL and glucose, then you are getting, if you ignore the weight, you are still getting what would be termed as the total effect of the QTL on glucose. If you adjust for body weight, then the meaning of the QTL scan is changed a little bit. If it is a mediator, if you adjust for weight, then it is the direct effect of QTL that on glucose that is not through changing body weight. So the meaning of the effect is altered a little bit. In this scenario, where they are controlled by different QTLs, you still get an effect of the QTL on glucose. The effect is conditional on the weight of weight at 11 weeks. So depending on what your goals are, you may want to adjust your interpretation or analytics strategy slightly depending on how you think the variables might be related. It's not always true that if you have another variable that appears to be causally connected, that you should adjust for it. I think that that's the main message. I would say that if you have body weight and glucose, it doesn't mean that you have to adjust for body weight. There is a case for not adjusting for weight. So let's do the scans here. I used RQTL here, and here is the scan for glucose, which is black, and body weight is in blue. In this case, I would say just eyeballing, other than on maybe the X chromosome and perhaps maybe the end of chromosome 1, all other, there is not much of an overlap between QTLs for glucose and body weight. So other than if there is a mediator, I think the only possible candidate for mediation is likely on chromosome 1. Any questions or comments up to this point?
So far, so good.
Okay. Okay. The next thing that we are going to go into is taking this idea of adjustment a little further. So what we have said so far is that if QTL are on different chromosomes, you might want to adjust for the QTLs, or if they are causally unconnected other covariates, you may want to adjust for them. So here is the standard linear model for adjusting for a covariate X. Now, if you think about this model, the standard way of describing this is a fixed effects model where you have a fixed effect for genotype, this beta, and a fixed effect for the covariate X, which is alpha. But what this model is also saying is that individuals with similar levels of X and G should have similar phenotypes. We are expressing that via a fixed effects linear model like this with kind of 2 parameters plus a variance. Now, if you had multiple X's, you would have many alphas and many X's, and you would extend this linear model and you would have more and more parameters. In general, if you wanted to control for genetic loci that are on all the other chromosomes, you could in principle do that by adjusting for all the genetic factors on all the other chromosomes by just adding other things. So for example, you might have a model that looks like this. You have a phenotype Y that is the sum of the effects of many different loci on many other chromosomes plus a random error. Now, this M, which is the number of other loci, could be very large. The main issue with this is that this becomes if you do this, in principle you could do this, but you will have a lot of parameters and it starts to become less efficient of a model. So there is a neat shortcut into doing that, and I'll try to explain the basic idea behind how you might do that. One of the reasons why this becomes important is, as we have seen earlier, that if you have if you consider a situation like this, or a situation where you have in a situation like this, if both of these are on different chromosomes, if you don't adjust for the other QTL, you have an unbiased effect. But as was pointed out in the comment, if you have correlated QTL, you might introduce a bias. Now, when you have a large multi-generational, multi-parental population, the random assignment of genomes to individuals is the randomization starts to be less perfect. And so it becomes important to adjust for other genetic loci other than the one that you are looking for. Okay, so that's the basic idea why you might want to have a model that has many different genetic loci that is being adjusted for. Okay, so let's assume that you have a model like this. Then the variance of y using kind of formulas for standard sums of random variables will be sigma square for this epsilon, the random error, plus the squares of the coefficients, the fixed effects coefficients, and basically the variance of the genotypes here. And assume that if you have coded the genotypes to have mean 0, then you just have the square of the genotype. So if you, again, just to simplify things, if you have like a -1, 1 coding of the genotypes and in a backcross or RI line, then this is always 1. So you have the variance is a sigma square plus the sum of squares of all the effects, approximately. If the loci are all unlinked or independent, then you can add this up like that. The covariance between any two individuals using a similar argument is because they will no longer share this random term, they will only, the random error term, they will only share the shared genetic effects using a similar argument will be the sum of the squares of the coefficients and this the covariance between the genotypes of the two individuals. And that can be expressed as this last term can be, this is basically the chance that if you pick two random locations on, if you pick a random location on the genome and you try to find out if these two individuals, I and J, do they have the same genotype? And because of the way we have constructed this population has to be identical by descent. And so that is the definition of kinship between these two individuals. So in other words, you have the variance is sigma square plus tau square, which is basically sum of the effects, the genetic effects, sum of squares of all the genetic effects. And the covariance is the same sum of squares of the genetic effects multiplied by the kinship coefficients. And here I have to make another assumption, which is in order to make the sum of the product be the product of the sum, I have to assume that the effect sizes of the loci are independent of the loci, which kind of seems reasonable. So the net import is that you can express this standard linear model by making with multiple loci. If you are willing to make a few assumptions about the loci being independent and the effect sizes being independent of the loci, then you can approximate this linear model with a lot of genetic loci as a mixed effects linear model of this form. So it has the same form as before, except I have, when I'm looking at a single genotype of interest, I have a single beta for that, and I have a random effect, gamma I, which has variance tau squared times this the kinship matrix, which is taking all these kinship coefficients and making them into a big matrix, and a random error that is independent, that is sigma squared times the identity matrix. So that's the basic idea of why we may want to use why we may want to adjust for genetic loci that are scattered all across the genome and are affecting our phenotype of interest, and and why we may want to use a linear mixed effects model in order to do that. Okay, I think I will go into how this looks like in practice in this cross, and depending on how we have time or questions, I can talk about kind of related things. I have another piece of a few other slides to expand on other things if people have questions or want to discuss anything. So now I'm going to switch to using another of Carl Broman's packages, which is meant for multi-generational crosses and many phenotypes, multiple phenotypes. So what I first do is, and his new package is called QTL2. So first thing I do is, he has a nice function, I'm very thankful for him, that converts the cross object from QTL1 to QTL2. So I first do that. Then I do basically insert a few pseudomarkers, calculate the genotype probabilities, and calculate the kinship from the genotype probabilities. One way to think about the kinship is basically it's like it's taking a product of the genotypes for any two individuals and adding them all up. That's essentially what the kinship matrix is. So for this F2 cross, this is what the kinship matrix, if you look at it as an image, that's what it looks like. You see a dark red along the diagonal and some splotches of red here and there. It's hard to say from this alone whether there is a whole lot of genetic structure or not. You can tell that you expect the diagonal to be dark because the kinship of every person with themselves is 1. So next, I think what we will do is I'll make a histogram of these kinship coefficients as calculated in this cross, and that looks like this. Sorry. Okay, so it looks, I would say, looks really nice, except that there is this long tail over here. Okay, the kinship between any two individuals, the expectation is that it should be 0.5, and it is almost exactly 0.5. Maybe there's a slight shift to the right, and there's a long tail. So it's not a proof, but just eyeballing it, I would say that the individuals here are related to the degree that we expect in an F2 almost perfectly, but there is a hint of population structure in that the distribution is like shifted very slightly to the right, not by a whole lot, but just by a little bit. So the first thing I did was I looked at the Eigen decomposition of this kinship matrix, and then I am going to look at the eigenvalues of the kinship matrix, and you can see that there is one really high eigenvalue, and all the other eigenvalues are close to zero. Actually, they should be all about half. That's the expected value. So if I take log 2 of the eigenvalues, I should see it a little more clearly. Let's run this again. So you can see that there are a bunch of very small eigenvalues here, and there is a smattering of larger eigenvalues, and one really large eigenvalue, which is expected. If this was a kind of a perfect F2 cross, then the eigenvalues would all have been half. So log 2 of those would have been -1. So that's not the case. So that's another way of saying that there is a little bit of structure in this data. Okay, so now what I'm going to do is I'm going to run a genome scan for our glucose trait with this linear mixed model. And then I plot the genome scan with using the linear mixed model. And just for reference, I'm doing the genome scan with a plain linear model. So as you can see here, there isn't much of a difference between the two ways of doing genome scan in this F2. And the main reason here is that, I was saying that there is a hint of structure, but overall, the individuals are close to what is the expectation for an F2 intercross, close enough that they can be considered exchangeable with each other, and therefore the linear model performs about as well as the linear mixed model. Now, as mentioned earlier, the idea of adjusting for these covariates, the other genotypes on the other QTL on other chromosomes, is that you want to soak up the genetic effects on loci that are not linked to the loci of interest. So going by that logic, the thinking is that instead of constructing a kinship based on the entire chromosome, one should exclude the chromosome of interest in the calculation of the kinship, when scanning a particular chromosome. So there is an option for that. It's called local, leave one chromosome out. And what we have to do in our QTL2 is first calculate a vector of kinships by this command called kinship calc with the option local, and then you pass that on to the linear mix model scan function, and then when it scans each chromosome, it's going to use the kinship you calculated using loci from all other chromosomes. And here is what that looks like. The dotted line is the one with loco, and the solid line is the one without loco, that is using the kinship using the entire genotypes. And in this case, except for chromosome 2, maybe a little bit of chromosome 1, there isn't much of a difference between these two approaches. So that's the summary of how we might want to use linear mixed models for to adjust for genetic covariates unlinked to the loci of interest. And you know, the topic of linear mixed models is more elaborate. There's a lot more to say. I've just skimmed the surface, and I would be happy to answer any questions, or if there are any comments, we can discuss. The main idea is that when you do a QTL scan, you consider the relationships between the various measurements that you have and how they might be related to each other. If depending on how they are related to each other, you may or may not want to adjust for them. You certainly want to adjust, if possible, for genetic covariates that are unlinked to your chromosome of interest, and linear mixed models provide a convenient framework to do so. I would be happy to answer questions or hear comments from anyone in the audience. And I have other slides. I can go into them if there are further questions or more other details. And maybe Carl and others who are in the audience could also chime in.
So Saunak, one question about the mechanism that could cause meiosis not to be a good process of randomization. So in the presence of meiotic drive, for example, in the collaborative cross, that was a problem, I believe, on chromosome 2. Could that explain, for example, even meiotic drive in an F2 intercross could rear its ugly head? And I'm just wondering whether that would be perhaps a cause for the slight distortion you picked up in Leia's data?
Yes, it could be. So I think that so it gets. There are many things to think about here. So
maybe we need to
first
meiotic drive for everybody. May not be a common concept for some people.
Why don't you describe it? I think maybe
during meiosis,
you can describe it better than I.
Assumed to be a coin flip, but there are rare exceptions where one particular haplotype or genotype has an advantage of getting into a sperm cell or an egg cell during the reduction divisions, and that results in distortions of the expectation of Hardy-Weinberg equilibrium and random everything. So basically, some violations of our expectations for meiosis of being pretty much a random process. And so that's what meiotic drive refers to, I think.
Okay, there's another thing which is related to meiotic drive, which is selection. I mean, I don't know how, I think there's a difference between meiotic drive and selection, but in this diagram here, I am assuming that there is no selection, and in some sense, I'm assuming there is no based on the phenotype of interest. And if there is, that again, that is which would be the case for certain human studies, I think where you ascertain individuals based on their phenotype, then it gets a little more complicated because you are selecting based on the phenotype. In the case of meiotic drive, you are selecting on survival, essentially. So you could consider this causal diagram to be correct, conditional on the individual having survived. So, I think that is maybe the caveat.
Got it. Yep, that makes sense to me.
So in particular, I think if there are loci on the same chromosome as what is being selected for, then you're depending on what you're looking for, what is the meaning of the QTL of interest is altered because you have a selected population.
I'll put in a link to a meiotic drive paper in the chat. But go on. Thank you. That was great.
Saunak, so far there's no questions in the chat, so people feel free to unmute yourself and ask a question or send it through the chat and I'll convey it.
I suspect I know where Saunak might have been intimating he was going to go next, to a population, I assume, with a strong substructure so we could see this in dramatic form. Is that true, Saunak?
That would be one direction. I think that there's a question in the chat. Okay.
There's a clarification question. Okay. Can you define eigenvalue?
Okay, so eigenvalue there is a technical definition of eigenvalues, and let me check here. Let me write in the technical definition of eigenvalue is if you have a matrix K and a vector X, and then it can be shown that for positive definite matrices as our kinship matrix usually is, they can be written in this form as if you multiply the matrix by a vector X, then you get the same vector X multiplied by a value lambda, and this lambda is called a eigenvalue. That's the definition, the technical definition. In this case, another way to think about the eigenvalue is if you think in terms of principal components, if you are familiar with that, then if you can think of the eigenvalues as being the how to describe this? Okay, as the amount of genetic variation that is being explained by a linear combination of the individuals. I think I'm not giving a good definition, and maybe somebody has a better, quicker definition. But if you're familiar with principal components, think of that as you're just basically doing something like a principal components for your genetic relatedness, and I think that's.
Steve has a follow-up question. Also, is there a way to formally distinguish the causal relationship between two different phenotypes, weight versus glucose?
So here that's a harder question. At least for the eigenvalues, there is a technical definition. Now, I drew this causal diagram for the possible relationship between weight and glucose. I always put an arrow between going from weight to glucose, with the idea that weight was measured before glucose, which was measured at 15 weeks. Weight was measured at 11 weeks. But it is possible that both of them have a common precursor, and it is possible that weight, how to say this, you cannot, maybe the glucose at 15 weeks is not necessarily causing the weight here at 11 weeks, but it may be possible that there is maybe glucose at 5 weeks is causing both a weight gain here and the glucose measurement at 15 weeks. So in that case, that would be the glucose at 5 weeks would be a confounder. So without additional information in general, it will not be possible to, from this data alone, to say whether the arrow goes from here to here, or whether there is something else. Of course, something at 15 weeks cannot cause something at 11 weeks, but there could be a common cause to both. But if they are both measured at the same time, then it is, without additional data, you cannot tell which way the arrow goes.
There's also an interesting example, Saunak, and relevant to some of your introductory thoughts about body weight as a cofactor or potential confounder. We've done a lot of studies of different parts of the brain and their volumetric control. And in that case, we usually want to make an argument about the specificity of a QTL modulating, say, the size of the hippocampus or the size of the cortex or cerebellum. And we do not want to confound it with body weight differences or even whole brain weight differences. So there we usually do not want to treat brain weight or body weight as a mediator because we're looking for a specific effect of the QTL in a particular suborgan of the brain. And so that's, there'll be some, but I think that highlights an example of where you want to remove body weight as a mediator because you're looking for that direct effect.
Mm-hmm. Yeah, I would agree. I think, the standard practice has been to add anything that appears related. And I think it is worth reconsidering that automatic practice.
Yeah, and to kind of continue on this, to get, we are planning sometime in the next month to give courses, a couple of webinars on mediation and causal analysis, both from a theoretical or semi-theoretical perspective, but also from a pragmatic perspective of how you actually implement this. I will participate in that, although I don't consider myself, I'm certainly not a statistician nor an expert in causal modeling, but I'm, I guess, a good guinea pig of a newbie coming into this, trying to make sense of it. In the book that I put up there, the Judea Pearl Book of Whys, so if you're interested in this, it's a way to get a head start on those topics. And yeah, Saunak has introduced these causal diagrams, I think, in a very useful way. And actually, even in his first discussion on QTL mapping, he did that. We're theoretically into the discussion section here. I personally would love to see if you've got anything for recombinant inbreds or collaborative cross or HS where the population structure is pretty dramatic. I don't know if you're prepared to go there right now, Saunak, but if you are.
I'm not. I tried, there were some glitches, which is why I could not complete that piece.
But
yeah.
I think the important thing to leave people with is that beautifully uniform F2 intercross matrix of relatedness, the kinship, the visualization of the kinship matrix. Imagine that now as a very intense mosaic. That is the common situation for an advanced intercross if it's not done quite right. For HS, if there's, again, if it's not a very large population, and for recombinant inbred strains where, Saunak said it well, basically any multi-generation cross is subject to that kind of intense kinship. And most human populations are subject to this kind of kinship. Remember that the Icelandic, when they started doing decode and genotyping Icelandic populations, they thought they would have very minimal kinship relatedness within Iceland because everybody would be exchangeable of Norwegian extraction. Well, it turned out that those Norwegian Vikings did little pit stops in Ireland, and so there was quite a bit of a mixture between Irish and Norwegian in the Icelandic population, which requires correction for the kinship. So
they also have very close relatives in there, so that's. So actually, this is a more complicated topic, but the eigenvalues tell you a lot about the structure of the population, so you can infer, at least in some way, the degree of sort of close relatives. So for example, here, if you, in this Eigen Decomposition, if you have two identical individuals, you should see eigenvalue corresponding to that pair of individual or a family. Or if you have a cluster of 5 individuals, then you will have eigenvalue, corresponding to that cluster or family. So that's, I think, yeah, maybe in a future time, I will be in a position to discuss that. So that was one of the reasons why I looked at these eigenvalues here. I mean, here there is I would say there is a hint of structure. It's not huge because you can see all the eigenvalues are at least smaller than 1, even if there is 1. The expected eigenvalue is about a half, which would be -1 here. So you can see that most of them are a little bit smaller than that. And there's even a few that are larger than that. So there is a hint of structure even in this population. It just happens to not matter a whole lot in the actual genome scan.
What does the sex difference look like? So the Y chromosome and the hemizygosity of the X chromosome should result in a low-grade kinship for all males and all females?
I don't think that is in there in the kinship. So it is, yeah, it is not calculated like that in here at least.
Okay, got it.
But otherwise, yes, there would be. That would be part of that kinship. Like, because yeah, I think it's only the autosomes.
Got it.